Hello friends, the topic for the day is biological classification. So we'll see all the kingdom Monera, Protista, fungi, plantae and animalia one by one. So today's topic is kingdom Monera. We're going to see about bacteria. So before that, if you didn't subscribe our channel, please do go and subscribe and press the bell icon for future updates. Yes, so we're going to talk about kingdom Monera. So kingdom Monera comprises of bacteria, and there they have split bacteria into two classes. One is Archaeobacteria and then is Eubacteria. So about the Archaeobacteria its characteristics and Eubacteria and its characteristic we'll see in the next class. Today we are going to talk about the common characteristics of bacteria, classification of bacteria based on its shape, and classification of bacteria based on its flagellar position. So first we'll talk about a common characteristic feature of bacteria. So the first feature is all bacteria are single-celled organisms. So single-celled, only one cell, okay? One bacteria you consider it as one-celled, unicellular organisms. Next, they are all prokaryotes. So prokaryotes are unicelled, single-celled, and also they lack rich nuclear membrane or a nuclear envelope. Alright, and next, based on its nutrition, the food, first they are all autotrophic. So most of the bacteria, they can prepare its own food. Next, some could be saprophytic, which means they feed upon dead organisms for its food. Next, and majority of these bacteria, they exist as parasitic forms. So parasitism means you harm somebody else and you get your food. So example, Helicobacter pylori. So most of us would have suffered from ulcers, right? So this is caused by Helicobacter pylori. They infect our digestive tract and they start utilizing the mucosal layer of the digestive tract as their own food and for metabolism purposes. Alright, and now we'll move on to the classification of bacteria based on its shape. So basically we have only four different shapes of bacteria: cocci, bacilli, spirillum and vibrio. First is cocci. So the term cocci, which means round spherical shaped bacterium. So this is how a spherical shaped bacterium, a unicellular spherical shaped bacterium. It is unicellular, it is spherical in shape. So that is cocci bacteria. Next is diplococci, which means these bacterium exist in pairs, like this, pairs. So you call it as diplobacterium. So they occur in pairs. Next is streptobacterium, which means that they occur in chains. So many diplobacterium bacteria, they occur in chain structure. So that is called as streptococci. Next is staphylococci, which means it resembles the shape of a grape. Clusters. So can you see many bacteria which are there in clusters? So cluster of bacteria is called as staphylobacterium, staphylococcus. Next is tetra. The word tet meaning four. Tetra means four. Four bacterium. So four coccobacterium, they are together called as tetra. They are always in fours. Next, we will talk about Sarcina. It is very simple. So, did you understand tetrad? The same way, two different tetrad bacterium, they join together. So, here is a tetrad bacterium. Beneath, there is one more tetrad bacterium. Alright, so there are 8 bacteria together. So, that is called Sarcina. So, next we will move on to bacilli. So the first is diplobacilli. So now what is bacilli? Rod-shaped cylindrical bacterium are called as bacilli. Now diplobacilli. So these bacteria, these rod-shaped bacteria occur in pairs and called as diplobacilli. So mostly they occur in pairs during the time of reproduction, that is binary fission, or during the time of conjugations. These bacilli, they occur in pairs. Next is streptobacilli. So for example, chain. So that's how we saw streptococci, right? They occur as chains. The same way, streptobacilli, they occur as a very long rod. They occur as chains. Next, we'll talk about palisade bacilli. You can also call it as palisade fins bacilli. And it is a typical, okay, now wait. Look at this, a zigzag structure where one pole of the bacterium is attached to the other pole of the bacterium. Only one pole of the bacterium, not both the poles. The other pole is attached to the next bacterium, resembling a zigzag structure. So this example is Corynebacterium diphtheria, which causes diphtheria. So next we'll move on to spirillum. So these spirillums are spiral shaped bacteria. So look at this, are they spiral shaped bacteria? Alright, look at this. Yes. Example, Treponema pallidum. Next is Vibrio comma shaped bacteria. So Vibrio cholerae. Look at them, they are comma shaped. So Vibrio cholerae. Next is Coccobacilli. So Coccobacilli, they are neither cocci nor a bacilli. They are oval in shape. Next we will move to Mycoplasma. So this Mycoplasma, they don't have a cell wall. They are very smaller than when compared to the other bacterium. Example, Mycoplasma genitalis. So they infect the urinary as well as the reproductive tracts in humans. So that's Mycoplasma bacteria. So now we are done with the classification of bacteria based on its shapes. Next we'll move on to the classification of bacteria based on its position of flagella. So flagella is basically a hair or a thread-like structure, so which helps in the mobility of the organism, the motility of the organism with the help of bacteria. So with the help of flagellum, these bacteria can propel themselves. Alright. So now we have many types and I'll show you. Okay. So first we have atrichous bacterium, which means a bacterium which do not have a flagellum, which is called as atrichous. Trichous meaning something related to flagellum. So atrichous meaning nil. There is no flagellum. Next is lophotrichous, which means there is a tuft of flagellum only towards one side of the bacteria. Look, there is a cluster of flagellum only towards one side of the bacteria. Next is amphitrichous. It's totally opposite to lophotrichous, where the tuft of flagella is seen on both the sides of the bacterium. Next is peritrichous. Example, Escherichia coli, E. coli, where the bacteria, where the flagellum is seen is scattered fully on the layer of the cell surface, on the cell wall. Next, we see monotrichous, where only one single flagellum on one side of the bacterium. Monotrichous, a single bacterium. Atrichous, no bacterium. Peritrichous, all over the bacterium. Lophotrichous, only towards one end. Amphitrichous, towards two end. So now we are done with the classification of bacterium based on its position of flagella. So next, move on to the session. First, we'll see about archaebacteria and then eubacteria. Now, the term archaea, which means old. They are one of the oldest bacteria which has been evolved. Okay. So you use the term archives, right? So which means old, something related to very old feature. So that's how. Before this eubacteria, it is always said to be believed that archaebacteria evolved first. Now archaebacteria, they all dwell in harsh environment. So harsh environment could be anything which is which is different from the moderate environment, that is harsh environment. For example, high salt solution in a very colder region, and also during hot springs, in volcanoes, during volcanic eruptions, and all these areas, these bacteria have hardest cell layer, that which makes them survive and metabolize even in such adverse conditions. Alright, and now you call this bacteria as extremophiles. So phile is something to dwell, alright, and extremophiles, which means they extreme, so they live in harsh condition, in extreme conditions, right? And now we'll see four important extremophiles. First is halophiles. Any bacterium that lives in a condition or in a solution or in any place that has high amount of salt content is called as halophiles. For example, brine solution. So we'll take a very simple example. Your mom is making pickles, right? So mostly in the doing process of pickles, what happens, they add pickles, they add these vegetables or fruit, whatever you want to pickle it up, inside a solution which is rich in salt content, right? High amount of salt content, that is called as brine solution. And these bacteria, they have the capability to dwell in such brine solution. So that's why even pickles are sometimes been spoiled by microorganism. Alright. Next is thermoacidophiles. So acidophiles, high acid content, sometimes a pH range of 1 to 4. So you call it as thermoacidophiles in terms of hot springs. So they are able to live in a habitat which is really hot. For example, near the volcanoes, volcanic eruptions, they are very easy to survive there. And these type of bacteria are called as thermoacidophiles. And methanogens. So these are the bacteria which produce methane. Alright, and they live in marshy environment. Now, these methanogenic bacteria, they almost dwell in cows and as well as buffaloes, okay? So mainly in their large intestine. So now these bacteria, they come out in their dung, and what happens is that when you ferment this dung in an aerobic condition, which means without the presence of oxygen, they liberate a gas called as methane gas. And this methane gas, like how you use normal petroleum as well as the other gases, right? Cooking gas. So instead of that, it is a most cost effective way to produce methane gas with the help of bacterium. Alright, and now you call it as biogas, which is produced biologically from the byproducts of microorganisms, and that's where these methanogens are being used, right? And that's it all about archaebacteria. Next, we'll move on to eubacteria. So the term eu means true, I told you in the last class. And this bacteria has two important characteristics. One is a rigid cell wall. Rigid cell wall means which has a well-defined cell wall, a proper cell wall. And then it has organ of motility. Motility means to move. So which means they have flagellum to move. Next, one distinguishing example we are going to talk in this eubacteria. There are many divisions in eubacteria, but in today's class we'll deal about cyanobacteria, which is one such eubacteria, right? And this cyanobacteria, you call it as blue-green algae. You call it as algae, but they do not belong to algae. Alright, that you have to remember. Sometimes in competitive exams, they might ask blue-green algae belongs to dash eubacteria, archaebacteria and algae. So you have to always choose eubacteria, never choose algae. Now next is, what is a very interesting fact about this blue-green algae is that they have a pigment, chlorophyll pigment, which is present in higher up plants, right? So plants do have chlorophyll with which they manufacture their own food with the process of photosynthesis. The same way, this blue-green algae, they manufacture their own food with the presence of chlorophyll. So they obviously undergo photosynthesis because they have chlorophyll A pigment, right? And now one more interesting fact about this blue-green algae. So there are many blue-green algae. For example, you have nostoc, you have anabina, and they have a peculiar organelle called as, or a storing organ called as heterocyst. In this heterocyst, they try to store atmospheric nitrogen. So we have nitrogen all around the atmosphere. So they try to capture this atmospheric nitrogen. They convert this nitrogen into ammonia, nitrites, nitrates, which is easily be able to take up by the plant roots. Okay, and these are called as nitrogen fixing bacteria. So these type of bacteria, they are always colonial, which means they live as colonies, they live together, and they have a protective outer covering, and you call it as a mucilaginous sheath. Okay, and next is where, along with its cell organelles, these bacteria have an organ called as heterocyst. Only in this heterocyst they capture the atmospheric nitrogen. Now what happens? They have captured this atmospheric nitrogen and they metabolize this nitrogen into ammonia, nitrates and nitrites. And now this nitrates and ammonia are being easily used by the plant roots and metabolized. Alright, and you call them as photosynthetic autotrophs because they undergo the process of photosynthesis like higher plants, and they are all autotrophs. Auto means they prepare its own, and trop means they eat by preparing its own food. So today's class we spoke about eubacteria and archaebacteria, and in next class we will talk about kingdom Protista. Thank you. And if you want to take a screenshot of it, you can take it. So yes, we're going to talk about Protista. So a very small history about this Protista, very simple history is that these protozoans are all single-celled organisms, right? They are able to move. For example, when you take amoeba under this kingdom, they are easily able to move. They have their own organ of mobility. So earlier, the researchers considered this amoeba into the kingdom Animalia because they can move, so they can do whatever, right? So that's why they are placed in the kingdom Animalia. Later on, they just found that they are nowhere related to kingdom Animalia and neither plants. So they created a separate kingdom called as kingdom Protista and placed them. Earlier, they were placed under kingdom Plantae, and few years later they were placed under kingdom Animalia. Then later, they got their separate kingdom, kingdom Protista. Alright, so that is all about kingdom Protista. So the first point is they are all single-celled organism, which means that they are just one cell. They just have a single cell. Next, they are all eukaryotes, which means that, so first the major difference between a prokaryotic and a eukaryotic organism is that prokaryotes do not have a membrane-bound nucleus. Neither the organelles of the membrane are not clearly present in them. But in case of eukaryotes, they have a well-defined nucleus, membrane-bound nucleus and membrane-bound organelles. Alright, so that is the simplest definition or the differences between prokaryotes and eukaryotes. Alright, yes, but now usually the organisms which are single-celled are mostly prokaryote, but only in case of kingdom protozoan like kingdom Protista, they are all single-celled but eukaryotic organisms. So when they ask questions like Protista are dash single cell prokaryote, multicellular eukaryote, single celled eukaryote, and none of the above. You can choose. They are all single celled eukaryotic organisms. Done. Next, they have their own organ of mobility, which means the organ which helps them to move from one place to another place. So you have three examples. Cilia. So I have paramecium here. See, they have cilia all over their body surface, the membrane surface. So that's how they move. So the organ of mobility for this paramecium is cilia. Next is pseudopodia. So we know amoeba, right? So they have pseudopods, false leg, with the help of it they move. Next is flagella. For example, when you take euglena, they have flagella. So this is their own organ of mobility. Now next we'll talk about reproduction. So they undergo both the sexual and asexual reproduction. So sexual reproduction is by cell fusion. So two of these cells fuse together and they form zygote. Okay. Next is asexual reproduction is by binary fission. So in my earlier classes I taught you the different types of asexual reproduction, right? So in that we saw about binary fission as well as encystation. So what is encystation? I told you earlier, where different three layers, proteinaceous layer is been developed around the organism during unfavorable condition, and now inside this there are many small spore formations. So once the condition turns to be favorable, what happens? These spore will break down, and this process is called as sporulation, where like baby organism, if it is amoeba, you have pseudopodiospores. So all the smaller organism of the parent cell has been detached. Alright, so that is all about the characteristic features. So we saw four important characteristic features of the kingdom Protista. Now kingdom Protista has five important groups. Alright, chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. So how to remember is that CD, SEP, CD and SEP. Alright, so chrysophytes, dinoflagellates, slime molds, euglenoids and protozoans. So now in today's class we'll talk only about the group chrysophytes, and in next class we'll deal about the rest of the four groups. Alright, yes, now first is group chrysophytes. Okay, and now this chrysophytes has two important organisms, that is diatoms and desmids. And now these desmids are also called as golden algae. Alright, so the name is just golden algae. Okay, wow. So which just doesn't mean that they belong to algae. Alright, so the name is just golden algae. So now diatoms. So what is diatoms is that these organisms, they have thick walls, okay, thick cell walls, and this cell wall is made up of silica, really high content of silica, which makes them to survive in the water body. It could be marine water body, fresh water body, and whatever. They mostly exist as planktons. So what is plankton is that a single celled organism which is able to move, and they are the food of most of the larval stages of higher animals. Okay, so for example you take tadpoles, the larval stage of a frog. So these tadpoles could feed on these planktons. Alright, so that's why you call it as plankton. So mostly kingdom Protista has huge number of planktons. And next, a very primitive feature of this cell wall is that they overlap on each other and they are compatible. For example, I just have this box, mini dustbin box. Alright, now what happens, you just imagine this is the lid and this is the bottom surface, right? And now just imagine how correctly this lid is able to fit onto the bottom surface. It's because the top is made in such a way that it is compatible to the bottom surface, right? The same way in this chrysophytes, two of the cells, right? The cell wall is so much compatible that two of the cells, they just join together and resemble like a soap box. So both of them just join this way. Alright, they are all single celled, but they just one cell joins with the other cell and they are all much compatible. So imagine this is one cell and this is the other cell. Now imagine how compatible the cell wall of the two, or like two different chrysophytes. Alright, so that's the concept. Next is, you call it as diatomaceous earth, which means that they have high content of silica in their cell walls. So once these organisms die, what happens? The silica content of the cell wall, they go settle at the bottom of the water surface. Now imagine this is happening for billions and billions of years. So the ground surface is fully filled with higher amount of silica, so you call it as diatomaceous earth. So that's all. Today we saw an introduction about kingdom Protista and the group chrysophytes. In next class we'll deal about the other 4 groups. So now today we're going to talk about 4 important groups under Protista. That is dinoflagellates, euglenoids, slime molds and protozoans. So first we'll see about dinoflagellates. So dinoflagellates, which means that 2 flagellas. So the organisms which are placed under this group dinoflagellates, they possess 2 flagella for motility. Alright? Yes. So now first we'll talk about cell wall. The cell wall is made up of cellulose plates, as in a plant tissue which is made up of plant cell which is made up of cellulose, right? High amount of cellulose. The same way, see, they are all confusing features. That's why they are first placed under plants, then animals, and then into a separate kingdom. So first, the cell wall is made up of cellulose plates. And now motility, as I told you, as the name suggests, dinoflagellates, they possess 2 flagella. So they have 2 flagella. Now pigments. So the important characteristics of this group is that they possess pigments. Alright. So they are seen in yellow, green, brown, blue and red color. So very good example of this organism which possesses red pigment is Gongalax. Alright. Now what happens? This Gongalax, they undergo rapid cell division. Alright. It could be binary fission or it could be multiple fission. So they keep on dividing. So mostly every organism, they take half to 1 hour for its own progeny to continue, for its own cell division, right? So now what happens? They keep on dividing. So as they divide, they form like a tiles. Now what happens? 1 becomes 2, 2 becomes 4, 4 becomes 8 and 8 becomes 16. So now what happens? As the time progresses, so these organisms, they divide enormously, and now what you see when you observe the water body, they observe as a huge red tile. Alright, they occupy most of the water body surface. So now what happens? They possess a toxin. Alright, and now this toxin is highly poisonous for the water dwelling organisms like fishes and the other organisms. Right? So yes, so now they undergo rapid division, they form tile-like structure and they liberate toxins which are toxic to the water dwelling organism. So that it's all about dinoflagellates. Next we'll talk about euglenoids. So a typical example for this group is euglena. Alright. Now first the cell wall. So now first in dinoflagellates, the cell wall is made up of cellulose plates. But in case of euglenoids, the cell wall is made up of pellicle, and this pellicle is highly rich in protein, and this makes its body to be flexible. Alright, now what happens when there is a small little space, or it has to swim in the water body along with the water current, so they are very easily able to twist and turn its water body. For example, see, that's how it is. So like example, this is a euglena and there's a water body. They are very easily flexible. So now you understand the term how they are flexible. They can easily able to rotate their body along with the water current. Next, is, yes, we saw about the flexibility. And now what happens in the presence of sun? For example, when they are on the upper surface of the water, they utilize the solar energy and they prepare its own food. So they are mostly autotrophic in the presence of sunlight. In the other time, when it is dark or when they are under the water body, when they are under the ocean, what happens is that there is no much of the sunlight could seep into the sea, right? So in this time, they depend upon the other organic components, the other organisms for its food, and they try to be heterotrophs. Alright, so they have two types of nutrition. So during favorable, they are all autotrophs. During unfavorable, they are all heterotrophs. Alright, so we saw about dinoflagellates, euglenoids. Next, we'll move to slime molds. Alright, so as the term suggests, slime molds are all slimy in substances. Okay, they produce a slimy substance, a mucosal substance around their cell body, which keeps them moist because most of them are saprophytes, which means that they depend on dead organisms. Example is that, for example, a fish dies and it is settling on the bottom of the water body. Now what do these slime molds go? They go and feed on the organic matter on these fishes and they metabolize themselves. Alright, so that's a very simple example of saprophytes. So they rely on dead organic matter. So organic matter is the dead living matter. The living matter which has been dead is called as organic matter. Right? And now don't confuse with the term plasmodium, because plasmodium causes malaria. I'm not trying to tell about the plasmodium which is causing malaria. Now what happens, these saprophytes, these slime molds, they rely on the organic matter. For example, there's a fish which is a dead fish. Now all of these slime molds, they aggregate on this fish, okay? So that's why you call it as a group. So now this group, like as in how in dinoflagellates, this Gongia-legs, you call it as red tiles. You denote this cluster of Gongia-legs into the common term red tiles. The same way these slime molds, they aggregate together, and this aggregation, this group cluster is called as plasmodium structure, which means these slime molds are in a clustered structure. They are all grouped together, right? And now what happens? They undergo sporulation. Okay, so simple. Next is our very favorite protozoans. Now these protozoans, you can divide them into 4 groups. So as the name suggests, the first is amoeboid, which means that they are all shapeless and amoeboid in shape, and a very typical example is amoeba. Next is flagellated. They have a single flagella as the organ of motility. For example, we have this euglena. Next is ciliated. They have cilia all over their body for their motility. Now what happens, these cilia, they react to the water current. Okay, now what happens is that as they move on the surface of the water, they try grabbing their organic food. Now this cilia helps the food to push it into the gullet. Okay, so gullet, you can call it as through an opening through which the food is been metabolized inside the paramecium's body. Next is sporozoan. They are all parasites. Okay. For example, we have this trypanosoma pallidum. So I guess this is this is trypanosoma pallidum. Now what happens, mostly this group, sporozoan groups, they exist as parasites and they cause diseases. So they are all spore producers. So at a single time they undergo multiple fission and they liberate huge number of smaller organelles. For example, if it's trypanosoma, they undergo multiple fission, and what happens, they liberate huge number of smaller trypanosomans through spores. Alright, so in this class we spoke about dinoflagellates, euglenoids, slime molds and protozoans. So